everyone, and I'm glad to see so many people here on a Friday. I'm really pleased to introduce Kenji Soto, who's going to be presenting his thesis research on habitat preferences of benthic organisms in the peripheral hydrothermal vent habitat of the Lau Basin. It says there's less words in the title. But that's the topic, and Kenji's done a really amazing job of overcoming some pretty overwhelming obstacles in pursuit of his thesis. For example, identification of the organisms. A lot of these organisms are undescribed and haven't been named, and so he had to become pretty much of a taxonomist in addition to everything else that he was doing to work on the community ecology in this poorly known region. His study is one of the first manipulative experiments that have been done at the deep sea around hydrothermal vents, and it's a pretty complicated effort to work with an ROV. And the most important thing, I think, is that you don't get a do-over. You get to do it once, and that's it, and that's your data. So Kenji has done a fantastic job with these very precious samples, so please join me in welcoming him to tell us what he has learned.
Okay, thank you everyone for being here. Let me quickly set up here. Okay, so today we're going to be talking about the habitat preferences of Lau Basin vent periphery invertebrates. First, I'm going to break down my talk. In the introduction, I'm going to introduce you to the study site and give you some background on the habitat differences that we see in the Lau Basin. Then I'm going to introduce you to my study question, and I'm going to introduce you to the different habitat preferences that we see and why these patterns might be existing. So this talk is divided into two parts. In the first part, I'm going to talk about the rocks that were collected to test the invertebrate community habitat preferences. And then I'm going to talk about how the rock characteristics were measured and how I went about analyzing the different characteristics. In the second part, I'm going to be talking about the fauna that I found, how they were collected, and how I analyzed those results. And then I'm going to be talking about the different reasonings behind what I found. And then finally, I'll be concluding with some overall major conclusions. So first, I'm going to orient you to the site. So where exactly is the Lau Basin? It's in the Southwest Pacific, and we have Australia and New Zealand down here. If you follow to the north from New Zealand, you see the Tonga Trench right here. Fiji and Samoa are a little bit to the north of that, and the Lau Basin is this area that's been boxed in red. All right, so why do we want to study the Lau Basin? So starting in 2004, it was established as a long-term study site because the hydrothermal vents in this area take place in a back arc spreading center. And this is important to note because previously most of the research that had been done at vents were only at oceanic ridges. So let's again take a look at another map. And we have the, sorry, wrong button. This button. Okay, so again, we have the Tonga Trench right here. And I mentioned that the Lau Basin takes place at a back arc spreading center. And what this means is that there's a area where new crust is being created on the back side of a subduction zone. So we have the subduction zone that's happening right here. We have the Eastern Lau Spreading Center, represented by this black line. And new crust is being created from the black line as you move to the east, and then from the black line as you move to the west. And as mentioned before, this is a long-term study site, so we do have 6 long-term sites that have been established. And today we're primarily going to be looking at the sites Kilo, Moana, Abe, and Mariner. In addition to the Lau Basin taking place at and a back arc spreading center. Another reason why it's worth studying the Lau Basin is because it takes place at a natural laboratory. And what I mean by that is that there are several gradients that occur in the Lau Basin, and this makes it an optimal location to set up a natural experiment. So for instance, one of the gradients that you see is that in the north, depth decreases from 2,600 meters to 1,900 meters. Sulfide decreases from 6 to 2 millimoles per kilogram as you move south. The bathymetry changes from a flat environment to a more peaked environment. And the extruded rocks that are being created in the spreading center change from basalt in the north to andesite in the south. And if you notice this red arrow right here, this is where the transition from basalts happens, where the transition from basalts in the north change to andesites in the south. Next, we're going to take a quick look at some images of the different sites. So this is from an image from one of the northern areas where we have these pillow basalts that have been extruded here, and you can see it's a very flat environment. And then here's an image from one of the southern sites, and you can see it's a very peaked environment. And you can kind of make out some of the rocks that were placed at the study sites that are represented by these little tiny objects here and here. So in addition to the physical, chemical, geological patterns, there have been some biological patterns that were also observed. In 2012, my advisor, Stacy Kim, published a paper describing how 3 different endosymbiotic mollusks arrange themselves in concentric rings around the hydrothermal vent sources. And what she discovered is that they were arranging themselves around the vent by their sulfide and temperature tolerances. And so we have the hairy mussel Alvinaconcha here in the closest ring, and the middle ring where there was the snail Iphomeria, and then in the outer ring the mussel Bathymodiolus. She also described this, while doing the study, that she noticed that there was this other area called the peripheral zone, represented by this blue area here, and they described this region as an area that was low on metals, having no sulfide. And the sulfide in this area is so low that it doesn't support chemosynthesis. The fauna that you see in this area, you do see some vent endemic fauna, but not as many as you would see closer to the vent. It is an area that it has higher primary production compared to the surrounding deep sea, and because of this, you do see an increased density of non-vent endemic fauna. And this area, it's highly variable. There isn't a set distance for how far the peripheral region extends, so it can range anywhere between tens to thousands of meters away from the hydrothermal vents. So we're going to take a look at some of the biological patterns that Stacy saw in the peripheral region. And one thing that they noticed was in the northern portion of the Lau Basin, so the area where you're seeing basalt-type rocks that are in the deeper parts of the basin, have higher sulfide, and in the flatter areas, the community here is dominated by primarily anemones. So we can see some large anemones here, and these tiny white little dots are smaller anemones. In the southern portion of the Lau Basin, so in the area where you're getting the andesite rocks, you see a community that's dominated primarily by sponges, and we can see these white little craters here are sponges. So why do we see these different communities of anemones in basaltic north and sponges in the andesitic south? One possible reason is the rock type texture differences, where the rock type is affecting the different fauna. And so one hypothesis is that the anemones are preferentially settling on the basalt, or they're somehow able to outcompete the sponges on the basalt substrate. Another possibility for seeing sponges in the south is that the sponges are preferentially settling on the andesite, or they're somehow able to outcompete the anemones on the andesitic substrates. Another possible reason that we're seeing these different communities is that rock type has no effect on the fauna settlement, but there is some sort of location effect that's responsible for the different communities. And so it could be either the depth, the sulfide, the bathymetry, or the temperature, or some other factor that's responsible for what we're seeing. All right, so what are some possible cues for why do we think that the rock type is affecting the different fauna? One possible reason is the difference in texture. So basalt is smoother in texture compared to andesite, and so one possible reason is that the basalt, that smoother texture is allowing for more anemones. And we do have a lot of evidence of surface texture affecting different larval preferences, and we've seen, it's been documented that the different settlement cues have been observed at scales from the centimeter to the micron level. And this pattern has been observed in barnacles, anemones, and polychaetes. There are also biological cues where barnacles and polychaetes have been observed to settle with conspecifics, or they're looking for bacterial films that they'll search for when they're trying to decide where to settle. And there's been studies showing that. Additionally, there have been chemical cues that have been determining fauna settlement. And polychaetes and limpets have both been shown to settle in the presence of hydrogen sulfide or potassium ions. All right, so in order to test if rock type has an effect on the faunal settlement, we set up a classical reciprocal transplant experiment. And so what we did is we have, again, the Lau Basin with our 3 different sites. We have our different gradients. We have our basalt rocks in the north and our andesite host rock in the south. And then what we did is we took the basalt rocks from the northern portion of the Lau Basin, and we put them in sites in the north and in the south. And then what we did was we also took the andesite rocks from the south and we put them in the north and the south. So how exactly was the substrate collected? In 2004, Charles Langmuir collected and classified 35 different substrates. And again, we have our map here. Just to remind you, we have our 3 different sites. In the north, we have high sulfide, deeper waters, and it's a flatter environment, and we're seeing basalts in an anemone community. And then in the south, we have, again, our sponges settling on andesite substrates. So in the north, at 2 different sites, he collected 2 different types of basalts, one a high titanium basalt and a low titanium basalt, and he collected 8 from the high titanium and 9 from the low titanium region. Then in the south, in the andesitic region, he collected 9 rocks from the Abe site, and then 9 andesites from the Mariner site. After collection, the rocks were cleaned and dried. Okay, so the rocks that were collected, they were originally collected for geological purposes and not as outplant substrates. So I wanted to test for differences in surface areas to test for potential biases. So like maybe one site was receiving more larger rocks compared to the other rocks, or compared to the other site. And here are just some pictures of the different rocks that were collected, and you can see that they're all different sizes and different shapes. And you can also see that they look like they're different in texture as well, and you can see that, and I tested for differences in substrate texture. So onto our rock type analysis. So for surface area, I used a 2-way ANOVA to test for differences in surface area by rock type and location placement. And then I used pairwise comparisons to test between the different locations, placement sites, rock type, and rock source. To test for differences in surface roughness, I use a MANOVA looking at the location and rock source, seeing if surface roughness differed by either of those two factors. So how exactly did I determine the surface characteristics? In order to get the surface area, I used the process of photogrammetry, and this involved putting a rock on a flat matted white stage with even lighting. I then would take a picture of the rock. I then would take another picture of the rock, and then I would rotate the rock, and I would keep taking pictures of the rock until I took about 70 different pictures. These pictures were then put into Autodesk's program Recap. And what Recap is able to do is it stitches all those pictures into one 3D model. So you get this cool little image of the rock. And Autodesk will also measure the surface area of the rock and give you a pretty accurate measurement of that. Additionally, Autodesk will also be able to export all the points that it used to create this model here, and it can be put into a script in MATLAB. So Ivano Aiello, he helped me a lot with this. He was able to help me get the surface roughness of the rocks that were collected, and using his script, we were able to get the surface roughness. So if we take a look at this figure here, this is just one of the faces of one of my rocks. Okay, so how exactly did Ivano's script help me calculate surface roughness? So I'm sure that many of you are familiar with the chain method for how you measure surface roughness. And what this method, just if you're not familiar, what this method is you lay a chain over some rocks, and then you compare the length of the chain to the planar distance, and you compare the distance of the length of the chain to the distance from the start of the chain to the end of the chain, and then you get your surface roughness. One of the problems with this is that it's very scale dependent, and so depending on the lengths of your chain, you're going to be getting the size of the lengths of your chain, you're going to be getting a different answer. And this is exemplified in this figure here. So we have a map of Great Britain here, and we have these different colored bars, and these are the lengths of different rulers that were used. So this purple ruler here was the largest ruler, and you can see the perimeters kind of look different depending on the different ruler size you use. In this figure, R is represented as the magnification, and as your magnification increases, your ruler is getting smaller. N is represented as the number of steps it takes for the ruler to get the perimeter of the Great Britain's coast. And one thing that you do notice is that as you're getting different sizes in your ruler, you're getting different roughness values, or your perimeters are different, so that's our roughness value. To avoid these issues of these problems that scale will give you, you can use the process of fractal geometry. And what fractal geometry does, it accounts for scale by incorporating your different scales that you use to measure your different length or your different perimeters, and it takes the natural logarithm of your magnification and plots it against the natural logarithm of your roughness. So in this case, it's the perimeter, and it creates a single value represented by the slope. So this is the blue slope right here. And getting a single value is important because it helps you compare multiple places while incorporating, or you can compare roughnesses across multiple places with a single value, and while you're still incorporating your different scales. And so just to compare, if we were to get a roughness with that was represented by a flat line, this would mean that the roughness that you've measured was an area that had zero complexity. If you were to measure a surface and you were to get the surface measured out to have a slope that was the same, that would mean that your surface was had the same complexity, but the amplitude of change was the same. So this, your y-intercept in this case is different, so the amplitude of change is greater on the red line. So a little bit, if we want to go a little bit into how Ivano's script is working, what it does is it uses a moving window to measure the surface roughness in different positions. And then it measures the surface roughness at different scales as well. So this is how we're incorporating our different magnifications. And for this study, I was using scales between 1 centimeter squared to 4 centimeters squared. All right. So let's see if surface roughness differed by placement location or rock source. So again, I looked at roughness at multiple window sizes. For the next couple of figures, I have the moving window size is going to be on the x-axis and this window sizes range between 1 and 40 centimeters squared. On the y-axis, roughness is going to be on the y-axis. And to compare the effects of roughness by source rock and location, I used a MANOVA test. So first, let's take a look at the surface roughness by our source rock. And if we see, we have this red and orange line that is consistently above the line, above the other two lines. And the red and orange lines are our two andesite rocks. So we have Abe andesites as red, Mariner andesites as orange. And then below those two lines are the blue and green line. And those are our two basalt rocks, the high titanium basalt and low titanium basalt rocks. So this confirms that andesites are rougher in texture than our two basalt rocks. And statistically, if we take a look that these rock sources are in fact, the roughnesses differ by rock source. Next, we're going to look at the surface roughness by location. And what you see here is pretty much just this blue line here, and this is the roughness of all the rocks that were placed at Mariner. And what you can't really see is a red line and a green line because those lines are underneath this blue line. And this is telling us that roughness didn't differ by location. So let's see how surface area differed by transplant location. Source. Okay, so now we're looking at surface area. So for this first figure, we're going to be looking at if surface area differed by transplant location. So we have our different locations here on the x-axis. We have our surface area of each rock in square millimeters on the y-axis, and we have our box plots of our surface area for the different locations here. We have means that are pretty similar. They're all around 3,500 or so square millimeters. And what we found was that surface area did not differ by transplant location. I also looked to see if surface area differed by transplant site, and it did not differ by surface area did not differ by transplant site. Next, we're going to look to see if surface area differed by rock source type. And so again, we have our surface area in square millimeters on the y-axis, and we have our different rock source types on the x-axis. We have our two andesites here on the left, and we have our two basalts on the right here. And we have our box plots of the different surface areas. And if we compare both andesites together, they had a mean of about 4,000 square millimeters, and the basalts had around their surface area was around 3,000 square millimeters, and they were significantly different from each other. If we look at how source rock, if the surface area differed by source rock, we see again that they were significantly different from each other here. And this is primarily due to the differences between the Moana basalt here, this purple box plot here, it being significantly different in size compared to the Moana high titanium basalts and the Mariner andesite blocks here. And it was not quite significant compared to the Abe andesites. All right, so did we avoid our biases? So for surface area, rock type, the surface area by rock type was different. Surface area by location was not different. Surface roughness in terms of rock type was different, and surface roughness did not differ by location. So we have successfully avoided biases by our different rock sizes. And so the different rocks that we had, they were evenly placed throughout our different sites. So again, we have our different sites here. We have our rocks of different shapes, sizes, and roughnesses, and then they're distributed evenly throughout our different sites. All right, so now that we know what the rocks look like and where they need to go, let me remind you of the question that we originally had. So why do we see anemones in the basaltic north and sponges and the andesitic south? So just to remind you, we have our map again. We have our 3 different sites. We have the transition here at the red arrow from basalts with anemones on them in the north. And then in the south, we have the andesite host environment with sponges in that environment. So one possible reason for these community differences is that rock type is affecting the fauna settlement. And then the other reason that we're seeing this community is that there's some location effect. All right, so placing the rock blocks. So once we had our rocks, we're then able to set them out in the peripheral zone to see if any critters were going to settle on them. In 2006, 35 rock blocks were placed at 3 different locations using the ROV JSON2. There's a picture of JSON2. Again, we have our map with our different sites. We have the ROV JSON2. So at our first site, Kilo Moana, in the host rock environment, there were 3 different sites that were set up where 2 basalts and 2 andesites were recovered at each of these 3 different sites. And then within each site, so within Kilo Moana 1, the rocks were between 1 and 5 meters apart. And then between the different sites, so from Kilo Moana to Kilo Moana 1 to 2, the distances were about 10 meters apart. So if we take a look, 80 kilometers away from Kilo Moana is the site Abe, again in the andesite environment. We had 3 different sites where 2 andesites and 2 basalt rocks were collected and were placed and recovered at each of those sites. Then 241 kilometers away at the Mariner site, again in the andesite environment, there were 3 sites where 2 basalts and 2 andesites were placed. In May 2009, 42 months later, the rocks were recovered. However, there were 5 samples that were unrecovered. So if you notice on the table, not every single rock was recovered. And this was due to the possibility that maybe some vents toppled over or one of the rocks might have fallen in a crack. All right, so how did we go about finding the rocks? I'm just going to show you just some pictures from the different sites. This is an image from Kilo Moana 2. So this is in the basalt environment. You can see we have one of our rock blocks here with a little ID tag. We have the tiny little white dots that you can kind of see are anemones, and then we have some larger anemones here. Next, we have a picture from Abe 2. These white dots here are probably barnacles. We have some mussels that are growing here. We have some spot lobsters here. And the ROV has a rock block in its arm. This is an image from Mariner, and you can see it again. It's in that high relief environment, and you can kind of see the different rock blocks that are represented by these little objects here. One of the interesting things too is these pictures come from the ROV virtual van, and you can go find these images whenever you want. They're available to the public. And you can also look through the notes that the researchers will leave behind. And one thing that you'll see in this particular note is that even with all this navigation that the ROV pilots have and that the researchers have, is that they still can't always find the different sites. So they were unable to find Mariner 1, and then they're aborting the search for Mariner 1. Here's an image of a successful recovery of one of the blocks. And we can see one of the blocks being placed into its own screw top jar. Sometimes crabs will get in the way of your research. And then sometimes, a researcher will start driving the ROV. So we have my advisor here driving the very expensive ROV, Jason too. Luckily, though, about, I think it was 25 minutes later, she hands the reins back to the pilot, and the ROV was undamaged. All right, so how did I go about processing these samples? So initially, after collection, the samples were fixed in formalin for 24 hours and then stored in 70% ethanol. Because many of the fauna that I was finding were sub-millimeter size, I had to use a microscope on a boom to scan each rock. And so what I did was I printed out sheets of images of each face of the rock, and then I put a gridded image on the rock, and I basically just scanned the entire face of each rock to see if I found anything. This particular rock is an example of a time where I didn't even need the microscope, so we can see a couple barnacles and a serpulid polychaete right here. And this was actually kind of rare to see macrofauna on the rocks. And primarily I was doing this at the 20 to 80 times magnification. I had help from my UROC intern, Hema Garcia. She helped a lot with scanning the rocks and going through some of the supernatant that the fauna were in. So after Hema and I would collect the different fauna, they were stored in ethanol, and they were organized by a major taxonomical group. I then had help from John Oliver and Valerie Lerb to help me identify the fauna beyond major groups. So they helped me identify some of the fauna to family, genus, and even sometimes species. And this was done with using a compound scope between 100 to 400 times magnification. All right, so how did I go about analyzing the fauna? Primarily, I used the statistical program Primer, and I was analyzing the data using densities because the rocks are of all different sizes, so I wanted to account for that. So I didn't use total abundances. And then I also used a fourth root transformation on my data just to allow for rarer fauna to have a greater effect. In Primer, I used the Permanova test to see which factor was responsible for the community. And then I used a analysis of similarity test to do pairwise comparisons between the different transplant locations. And then I used a SIMPER analysis to see what the fauna composition was and to look at the similarity that each site had to itself. So next, let's take a look at some of the community analysis that I did. So first, we're going to take a look at a non-metric multidimensional scaling plot that was done in Primer. So if we take a look at this figure here, we have a bunch of different shapes with a bunch of different colors. In this diagram, the salts are represented by circles, and the sites are represented by triangles. So we can see the triangles are kind of distributed all over the place. The circles are also kind of all over the place. Also, our different locations are being represented by different colors, where Kilo Moana is represented by blue shapes, Abe is represented by red shapes, and Mariner is represented by green shapes. And one thing that you notice in terms of the different locations, green and blue are kind of also all over the place, but red is very close together. And so this means that the rocks that were placed in the Abe location are fairly similar to each other. Next, let's take a look at the Permanova results, and we'll see that the only factor that was significant was the location. So this means that location had a significant effect on the different communities, and the fauna organized themselves by location. Site was not found to be significant, and neither was rock type or rock source type or the interaction between rock type and location. And because location was the only factor that was significantly different for the fauna composition, I'm only going to be talking about the results for each location. If we look at the analysis of similarity results, we look at and we see the pairwise comparisons between the different sites. We see that Moana and Mariner are not significantly different from each other in terms of the community analysis, but Abe is significantly different from both Moana and Mariner. So there's something going on over at Abe. So let's take a look at some histograms from the different fauna that we found. So what you notice is that there are higher densities of fauna occurring at Abe. So we have our different locations on the x-axis here. We have Abe here, Moana here, and then Mariner here. Fauna density is represented by individuals per square meter on the y-axis. And we have different fauna groups represented by different colored bars. So we see that copepods, represented by this blue bar here, are significantly different at Abe compared to the two other sites. Polychaetes occur in significantly higher densities compared to the other two sites. Mollusks and barnacles also occur in significantly higher densities compared to these other two sites. And the other groups are not significantly different from each other, comparing the different sites. Next, if we take a look at the similarity percentage analysis, so what we're going to be looking at is just the top 90% most common fauna. And what I did is, so we're going to be looking at different fauna, and they're going to be highlighted by a major taxonomic group with these different colors. So first, if we take a look at this first line here, Moana, and it says average similarity 15.41. So this means that all the rocks that were placed in Moana, they're about 15% similar to each other here. Abe is about 40, all the rocks placed at Abe are about 41% similar to each other. And then Mariner was about 34% similar to each other, all the rocks placed there. Next, we're going to be taking a look at how these tables are being, how they break down. So first, we have one column of our different taxa that are going to be highlighted by our different fauna groups in this column. And then in this next column, it's going to be our average density. And then next, it's going to be a contribution percentage here. And then this percentage is just the total contribution that a particular fauna is having towards this top 90%. So first, we're going to take a look at the copepods. And we see that they are one of the most common fauna that are occurring across all sites. And they make up about 54% of the fauna at Moana, 35% at Abe, and about 65% of the fauna over at Mariner. Next, let's take a look at the polychaetes, and they were the next most common fauna that I found, and you can see they make up about 23% fauna at Moana, 31% of the fauna at Abe, and about 8% at Mariner. Next, let's take a look at the mollusks and the other crustaceans, and you can see that they don't really occur in that high densities at these two, at Abe and Moana, but the other crustaceans occur in moderate densities at Mariner. One thing that you'll notice is, actually that you don't notice, is that you don't see any anemones or sponges, and this is kind of interesting because these were the two fauna that kind of brought up this question, and not finding either of these fauna is interesting because we can't really make any determinations of whether rock type is actually affecting these two communities because we didn't really find any of them. But we can say that rock type did not have an effect on the fauna that we actually did find. All right, so next let's take a look at Moana. So as previously mentioned before, we have our map with our different sites. Moana is here in the north in the basalt region, and it has anemones on it. It is deeper, it is higher in sulfide, and it's in a flat environment. Okay, so although we saw high concentrations of sulfide in Moana, but we also saw low densities of fauna. And this is interesting because when you think about hydrothermal vent ecosystems, they're primarily driven by the presence of sulfide. So where sulfide is the necessary component that allows for chemosynthesis for free-living, mat-forming, and endosymbiotic bacteria that form the base of the food web. So you would typically expect to see, in areas where you're having higher sulfide concentrations, you would see higher densities of fauna. However, one reason why we think that we're seeing lower densities of fauna is the emission type that's occurring at Moana. So in this area, you're seeing emission types of where there are point source emissions where you're getting a single hydrothermal vent and the fluid is rising up to the surface, and it's kind of the overall area where the fluid is being distributed is limited. So the fauna here don't have as much access as they would if you were not getting these point source emissions. Next, let's take a look at the Mariner site. So again, to remind you, at Mariner, the sulfide concentrations were lower. They were actually in a moderate concentration. And this area was in the andesitic environment that had sponges on it. It was shallower and flatter as well. Or sorry, it was in the peaked environment. Fauna here were found in moderate abundances or densities. And one of the reasons we believe this is because of the emission type of the hydrothermal fluid is in a diffuse manner, where the fluid is going through the crust and it's diffusing laterally in a large area. So even though you're getting lower concentrations of sulfide, the sulfide is still spreading out in larger areas, so this is allowing for more sulfide to be available to the different fauna. Just some other notes. So I did lie to you a little bit. I did show you this sulfide decreasing as you move north to south. Actually, in Mariner, we were seeing some higher sulfide concentrations, and this is because the Mariner area is geologically active, which we have evidence of this because we lost some samples due to the vents toppling over. And this geologic activity is also another reason why we think that we're seeing higher sulfide in this area is we saw some larger terabellid polychaetes. So this could mean that the increased levels of sulfide are allowing for larger fauna to live and grow. And also the geologic activity that we see could also be responsible for intermittent venting that's happening in this area. So next, let's take a look at Abe. So just to remind you again, so sulfide concentrations were low at Abe. They were in the andesite host environment, and they had sponges on them. They were in depth was in the lower depth areas, and lower sulfide and more peaked environment. Fauna, however, at Abe was higher. And again, we think that this is probably because the sulfide is being distributed in a diffuse manner. So you can see here, this is the hydrothermal fluid, and it's being distributed over a large surface area that's allowing for more fauna to live. Just some other notes that we had. So because this is a deep sea environment study, we were only able to get sulfide measurements at 2 points, and we couldn't go out to the site and get a continuous measurement of the sulfide. So we can't really necessarily say that, yes, sulfide was higher at these sites. But however, we can use some of the fauna that we saw to make some hypothesis for that the sulfide was actually more available to the fauna. So what are some of the hints that the fauna are giving us? So as I mentioned before, this is a deep sea study, so we don't have a lot of chemical data. Stacy mentioned earlier that many of the species, they're poorly described, and this is even including some of the near vent fauna, and even the, so if you can imagine the lack of information about the near vent fauna, there's even less information about the peripheral area fauna because most of the research that occurs at vents is actually in the near vent environment and not necessarily in the peripheral region. So we're going to take a look at some of the vent endemic fauna that occurred at Abe. So these fauna are the ones that there's actually more information about. And one of the interesting things was that at Abe, we were primarily seeing these vent endemic fauna. So these fauna are the ones that you're normally going to be seeing closer to the vent, and these ones are also more common at Abe than any other site. So the densities that these fauna were occurring in were higher than the other fauna. So if you recall, I mentioned the mussel Bathymodiolus. This is one of the symbiont-containing mollusks that my advisor, Stacy Kim, was studying. And we only found these at Abe. And because this is a endosymbiont-containing taxa, because it was only at Abe, we hypothesized that it was able to survive here because there was more sulfide at this location. The barnacles that I found were also only at Abe. And one hypothesis why, or more evidence for why this barnacle, that we think that the barnacle is providing evidence for more hydrothermalism, is that I saw some possibly pedunculate barnacles, and there are some pedunculate barnacles that occur at the Lau Basin that are known bacterial farmers that will grow bacteria on their cirri. I can't determine if this is one of those barnacles or not. Also, I did find a few limpets at Abe, and they were found in higher densities compared to the other sites. And the limpets that I found are known bacterial mat grazers, so they're obviously in areas that you would find more sulfide to support their bacterial eating. Also, I found I found one genus of copepod and then a family of other copepods in higher densities as well. And these copepods are found commonly in the mussel washings, so the Bathymodiolus mussel washings. So those, again, they were in environments of higher sulfide. And I also found higher densities of some polychaetes that are known to be in the near vent environment, primarily polychaetes from the family Amphorettidae and Serpulidae, and we have an amphorettid polychaete right here. All right, so some other studies that are that help inform this one. In 2009, Benjamin Group published a paper about the near vent communities at the East Pacific Ridge and East Pacific Rise. And what he was doing, he was testing different types of substrates like wood, biogenic tubes, and different shells, and seeing if these different types of substrates were affecting the fauna. And what he found was that substrate had no effect on the fauna, but sulfide concentrations were the main driver of the communities. And then, as previously mentioned before, my advisor Stacy Kim and Camille Hammerstrom in 2012 published a paper describing how the different fauna are arranging themselves around the sulfide, and they were looking at the near vent communities in the Lau Basin. And then in 2016, Arunima Sen published her thesis about the peripheral communities in the Lau Basin, and what she was finding was that there were also distinct communities in the north and the south, and then the two communities were also differing primarily by sulfide presence and not necessarily the different rock types that we find in this area. Okay, so what are some possible ways that we can make my study a little bit better? I think one thing we could do is to get more chemical data. So if we could actually get some sort of continuous measurement of sulfide, we could actually confirm if there was more sulfide or not. Another thing would be is to possibly use like a laser scanner to get a more precise value of roughness. And then another thing that would be beneficial is to get a higher level of identification of the different fauna that we found. Okay, so what did we find? Just to remind you of our question. In the Lau Basin's peripheral hydrothermal vent zone, why do we see anemone communities in the basaltic north and sponges in the Pacific south? Is it because of rock type? Not that we have found. Is it because of location? Probably yes. And then that's it. All right. Okay, so I just want to give a little shout out to my different advisors. Just say thank you, especially and say Stacy, thank you very much for just being very supportive and just always being there and being patient and just being there when I needed you. And thank you, Ivano, for also being very accepting and explaining rugosity to me 50 million times and me still not getting it entirely. But thank you. And then thank you, Kenneth, just for being just a positive force in my life. I'd also like to thank my parents just for supporting me and thanking my dad just for continually asking me how my thesis is going, even if I didn't like it. I'd also like to thank the greater Moss Landing community just for being a very positive place and just always being there for me. And then a special shout out to the Frisbee community in the Monterey area. I don't know if I could have gotten through this program without being able to play Frisbee every week. Any questions?
Jim. So, remind me how long the time frame was between when they were set out and when you recovered from the
42 months.
42 months. Do you think you would have got a different answer if you would have left them out longer?
It kind of depends on the stability of venting. 42 months is a long time. There's evidence that the Lau Basin is fairly stable in terms of venting on the order of decades. So I don't really think so. Maybe if they were left out for 10, 20 years maybe, but another 5 years, I don't think that we would see it. I think if they were moved maybe closer to the vents or something like that, then maybe. But I think you have to leave them out for a significantly longer amount of time.
So related to that, what's the life history of the anemones that might be impacted by time?
So I did find. So actually, you can see here, I did find one little anemone here. So at some point within those 42 months, one new anemone did recruit or settle on the rock. So probably a couple years.
But do we know very much about the life history?
Yeah, I haven't read this paper in a long time, but there was a paper that was published describing the physiology of, I think, 3 or 4 different anemones that are found in the area. But since I only found one, I didn't really look into the life history of the anemones.
Paul. Yeah, sort of a similar question. I'm curious, the time issue and also maybe bacterial communities that may have formed on those rocks.
Mm-hmm.
Is that a thing you can observe, like a bacterial biofilm starting to form on the rocks? Did you see that on rocks, like from your video footage of ones that you're recovering versus ones that are out there already? Does that explain some of the difference in composition of animals that are growing on them?
So I did not take into account the different bacterial communities because they were given to me, afterwards. They were, fixed in formalin and then preserved. So I didn't really account for the different bacterial communities. And then to look at the actual bacteria growing on them, you would have to look at the ROV surveys, and I didn't look at that. So I'm just trying to get hints from what the different fauna that I found actually living on the rocks.
Similar question, but related to the sponges. So did you find any sponges at all?
I didn't find any sponges, but I did find some sponge spicules.
Just fill out history of those sponges and how quickly they reproduce. Would you even see sponge settlement on the rocks in that period of time?
I'm not sure about settlement, but I think we would, we would see growth and decay because there was evidence in the Aronima 2016 paper where they observed one sponge growing and then dying. I think it was at one height at one time, and then maybe it was smaller later when they went to go do the survey later. Yeah, so I think that was over like a 3-year time span. Yes. I saw some other hands back there.
So following on the not observing any differences in settlement of sponges and very little in the anemones, what do you think are some reasons that you might not have seen any recruitment for these two species, where you did see recruitment for other species?
I think we were just seeing that, rock type is having a significant effect, or it's not having a significant effect on the fauna, and this was the primary driver for those two communities. Maybe that they're just it's not in the optimal environment for these two fauna. We have seen, as I showed earlier in the other slide, the different studies. Pretty much hydrothermalism is the main driver for these fauna. And so just maybe the hydrothermal or the sulfide community just wasn't optimal for the anemones or the sponges to.
Good at one point.
Yeah, so.
And then it was when everything recruited, and then they were just kind of hanging on. But
Yeah, so maybe there wasn't like a recruitment event or anything like that. It's hard to say that since we don't have accurate time series of the amount of sulfide that was in the area. Yeah. Jake?
I saw in your fauna tables that some of the species, they're probably just undescribed,
Yeah.
right? Copepod 1, copepod 2, copepod 3. Do you have any plans to describe those?
Well, it's not necessarily that they weren't described, or maybe they're not described, but I couldn't figure out how to identify them, and I couldn't find any literature on them, so maybe they aren't described. I don't know. I don't have any plans at the moment. There was one polychaete that I know for sure I'm pretty sure it hasn't been described because it's from a family from Glyceridae, and I haven't found any documentation of them. Is it in this picture? It's not in this picture. It's in another. I have another picture on here somewhere. But yeah. Do you want to become a copepodologist? Oh, also there is one researcher in the Netherlands that's doing a lot of genetics and species descriptions on hydrothermal vent copepods. So maybe they're on it.
All right,